I'm really interested in the fusion of art and science. I really like to capture the beauty of plant biology, some of the things that the public don't see that we see regularly on a daily basis as scientists. I remember I had a friend that worked, whose parents worked for Monsanto, and he was telling me, oh yeah, you can change the color of a tomato through genetic engineering or make the tomatoes bigger. I was like, nah, man, that's not real. That's magic. You can think of a plant cell as being like encoded by 25,000 genes, like the blueprint for the cell that include proteins that are bumping into each other and causing chaos, within the cell. Somehow it still manages to create the plant on the macro scale, which is quite incredible. But to engineer that chaos is really difficult. One gene circuit that I've made, it's some genes from beetroot. So beetroot produces this really strong pink pigmentation called betalain. And there's three genes that underpin the synthesis of this pigment. You can put that into your plant of interest and it will turn the plant pink. When I usually do public outreach in shows, for example, people have never heard of Arabidopsis, despite the fact that it's the most studied plant in the entire world. If I see a phenotype, like the effect of a mutation on the appearance of the flower, if it's quite nice, then I add it to my flower design Illustrator file.
Yeah, I can imagine Nick going through his studies and what do we have on the menu? Okay, this looks
Yeah,
nice.
yeah. It's like a wizard's cookbook for plants. It's amazing. If I wanted to genetically engineer a new beautiful type of rose, this is going to take on the order of decades, right? But as you can see, just within a year and a half, you can really transform Arabidopsis into something completely different.
Can you use an animal gene and put it into a plant? As an example,
Absolutely. Yeah.
yeah?
Yeah.
Really? So,
For example, we make melanin as a gene called tyrosinase that converts tyrosine to melanin, this dark pigmentation. Kind of goes from light brown to black. I kind of had a wild idea. I was thinking of putting my tyrosinase into the plant to make it have melanin and to be like a black gothic flower. And then it's like I am a part of my artwork and my gene replicates with my artwork.
do-
And then once I'm gone, like in 500 years, but my plants live on, my little gene is in there and it's like melded with my artwork and my legacy.
oh my God,
Yeah.
this is brilliant.
Some are saying I'm gonna start the next zombie apocalypse, or that like I hate nature because I'm trying to design it. And then other people saying this is like the most beautiful art project ever.
Hi, Nick. Thank you for being here.
Thanks for the invitation.
Oh, yeah. Actually, I've been wanting to interview a plant engineer for quite a while. At some point, I stumbled upon, I think, Light Bio with
Hmm.
this glow in the dark petunias. And I
Yeah,
was like, oh my God, what is
yeah,
that?
very cool.
Yeah. And did you study together with the guy who started the company?
No, no, no. But I actually got to know him a little bit
Mhm.
after he started it because I find genetic tools and plants super cool, like you do. So I was interested in getting my hands on the plasmid that he had made for autoluminescence in plants, especially because I do time-lapse imaging. So I had reached out to him after the paper was published, asking for the plasmid in some of the plants. I actually asked for all the plants that they made, which was like Nicotiana, Petunia, Arabidopsis, and Poplar. And he's like, well, I can't give you all of them. My poplar's a tree. But I got some of them and yeah, I did some time lapse with them. It's even on their website now.
Oh,
But yeah,
beautiful.
Karen Sarkisian's the guy that made this, invented it. And he's a really cool guy in Imperial College London.
Let's start. First
Mhm.
of all, I would ask you to briefly introduce yourself for our audience because we
Mhm.
started right away.
Yeah, yeah, yeah. So hello, everyone. I'm Nicholas Desnoyer. I'm originally from the US. I lived there until I was 24. It's where I did high school, bachelor's, and master's. But then I moved to Switzerland to do my PhD in plant biology. And now I just recently moved to the UK where I'm doing kind of this flower engineering project that I think we'll talk about today. But I'm really interested in like the fusion of art and science. I really like to capture the beauty of plant biology, some of the things that the public don't see that we see regularly on a daily basis as scientists.
Yeah, plant biology is really fascinating, especially when you are representing it in this beautiful artistic way with time lapses and everything.
Mhm.
So it's mesmerizing. But where did your fascination with plant biology start?
Hmm. I probably started earlier than I originally thought. My parents, they have some property in the countryside in Missouri. So I spent a lot of time outside, and there were some times that I remember like dissecting plants and like pulling apart the green smushy stuff. And I was like, what is this? Like, how is it self-assembling and all of that? So those questions I think came on really early. I also remember I had a friend that worked, whose parents worked for Monsanto, and he was telling me, "Oh yeah, you can change the color of a tomato through genetic engineering or make the tomatoes bigger." I was like, "Nah, man, that's not real. That's magic." And then later in college, yeah, I started to take some plant biology courses, got into a lab, and that's what really hooked my fascination.
This is so cool how our childhood memories they
Yeah.
shape our future.
Totally, totally. Yeah, I think you can track back most people's passions to like really early childhood memories. And actually, if you want to like unleash your superpower, you should probably look back at like what are the core things that I was curious as a kid that persist to today.
This is a great advice, certainly. I will need to think about that. I think I was reading encyclopedias all the time.
Yeah.
So, what was your first plant engineering project and when it happened?
So I guess it depends on what you define as like plant engineering. It's kind of a broad term. The first thing that really captured my fascination was something that we do all the time in plant science labs, which is fuse genes to genes from jellyfish that are called fluorescent proteins. It's a way to track protein localization within cells. So essentially, you add a fluorescent protein tag to this protein, and then with a confocal microscope or fluorescent microscope, you can see where that protein is in the cell. But to do that, you need to take this gene and put it into the plant genome. And so in that way, you've kind of rewired the plant genome to produce, express this synthetic protein that you've made. So that was, I guess, the first kind of tiny project that was back in my master's. But I think plant engineering would be more like really trying to control its form and patterning and maybe response to certain environmental stimuli in a robust way. And in that sense, I would say it's, yeah, probably this flower engineering project. But it kind of, I think this word engineering is kind of loose because engineering in my eyes is like everything it's you do is on purpose. It's with intent that you say, I'm going to mutate this gene and it's going to cause this effect. And in engineering, it needs to be like that. Whereas biology, it's quite messy. You don't know how the plant's going to interact. So what I essentially did is look at many nice phenotypes that have been described before that occur when you break certain genes or mutate certain genes. And I put these all together alongside some transgenes that change the color of the flower. And so the end result is a flower that looks morphologically much different than the original, has a different color. Um, in a way, I would call this, yeah, engineering.
Tell me how, in a nutshell, this works. So, what do you
Mhm,
do? I mean, most of the food that we're eating right now is
mhm.
practically engineered. Yeah, because
Yeah,
if you take a look at wild fruits or vegetables, they even do not resemble anything that you buy at, I don't know, the
yeah,
supermarket.
yeah.
Yeah. So, but how you do plant engineering? So more advanced version.
Right. So it comes back to how you define engineering. For me, engineering requires intention that you say, I'm going to make this exact phenotype, and then you do it. Whereas what we've made with modern crops, I wouldn't necessarily consider genetic engineering, more just selection, artificial selection. The classic example is modern corn. It came from teosinte a couple thousand years ago when we started to find the phenotypes that we liked in mutant teosinte that made seed coat more edible and made the fruit larger. And so over thousands of years, you can select for the traits that you like. But what's causing those traits is mutations in the genome, largely caused by just accidents happening in the cell, particularly how the DNA replicates. And so I wouldn't necessarily call this engineering. For me, engineering is again with this intention. And so what I'm doing is precisely mutating genes that we know the effect. and combining these. And so you can think of like a plant cell as being like encoded by 25,000 genes, like the blueprint for the cell that include proteins that are bumping into each other and causing chaos, within the cell. Somehow it still manages to create the plant on the macro scale, which is quite incredible. But to engineer that chaos is really difficult. And so what I think is really cool and what is really an engineering approach is adding genes on top of those 25,000 genes that don't necessarily bump into all of those parts. They kind of do their own thing. And you can predict how they'll work. So for example, one gene circuit that I've made, it's some genes from beetroot. So beetroot produces this really strong pink pigmentation called betalain. And there's 3 genes that underpin the synthesis of this pigment. You can put that into your plant of interest and it will turn the plant pink. And so I made a small genetic circuit where you can add a hormone to the plant, and then this induces that pink pigmentation. And these kind of DNA parts, they're not embedded within this chaos of all the interacting parts that have evolved together for millions of years. What I'm really interested in with the genetic engineering aspect is adding another level of sophistication on top of what's already there to really control the plant morphology and patterning from like a top-down approach. If that makes sense. Sorry.
Yeah,
It was
that makes
quite
sense.
complicated.
I'm super interested. First of all, so it's like an update, yeah, like on top of that, built on top of original biology of a plant. And
Yeah.
since you add only one circuit, it's more controllable than if you
Yes.
would
Yes.
add it to the all this mass
Yes.
that
Yes.
is there.
Yes. You can kind of predict better what's going to happen. Yeah.
You put it in the plant. What do you do for
Yeah,
that?
that's always the question. So there's a few ways that you can do this. The most common way, at least for Arabidopsis, the plant I work with, is to use agrobacterium-mediated transformation. So you create the DNA that you want. So we can talk about that as well. And you put this into a bacteria. You can put it into the bacteria through several different ways as well. What we do is a thing called electroporation, where you shock the bacteria with a small amount of voltage, and then it kind of freaks out and takes up the DNA. But this agrobacterium is really like the vessel for delivering the gene now into the plant genome. As agrobacterium in the wild naturally does this, it infects plants and creates what's called a crown gall. So it inserts genes into the plant genome that essentially make the plant make a home for the agrobacterium that also kind of feeds it. You've probably seen these crown galls on like trees. They kind of look like tumors. Some really smart scientists, they removed kind of the virulence genes. They removed these virulence genes that create this tumor and instead just kept the genes that allow for the insertion of the gene into the genome. And so we put these genes into agrobacterium and then we actually dip the bacterium into the flowers. And some of the bacteria, while the early flower bud is developing, can make their way down into the reproductive tissues where they can transform the cells that lead to the egg cell. And this will be like a stable transformant. So the gene gets into these germline cells, and then in the next generation, all of the cells within the plant carry this gene.
When it's stable, yeah. So
Stable, right?
your beetroot flower will reproduce and
Yes.
stay pink.
Yes. As long as it
Wow.
doesn't get silenced, which is another aspect called epigenetics, which happens
Yeah,
often.
RNA, yeah. So it can be silenced. It still can fade at some
Yeah, right.
point.
Yeah. Yeah. So the plant has mechanisms for recognizing foreign DNA. Or if something's like expressed too much, then the plant recognizes this and can silence it. Yeah. So then they wouldn't be pink anymore, which does sometimes happen.
You said that you put it on top of existing DNA of a plant, but
Mhm.
how? I mean, why
Mhm.
it
Yeah.
does not plug into the original DNA?
Okay, yeah, maybe I was a bit misleading with that. So it does. So Arabidopsis has 5 chromosomes. Within these chromosomes is the entire genome. And so each chromosome, gosh, I don't know, is maybe, I don't want to say something wrong, maybe 20 megabase pairs, so a million letters. And this tDNA, this gene that
Mhm.
comes from the bacterium, it will be sandwiched somewhere kind of randomly within this chromosome. And depending on where in the chromosome it lands, it may have different expression levels. because some parts of the chromosome are more accessible than others. Like they can become very densely packed into what's called heterochromatin. And then that gene I put in probably won't be expressed or produced. So it does still get put in alongside the other genes. I just mean that these parts, a lot of them are coming from viruses or from bacteria where they haven't evolved along with this complex network of genes. And so in a way, you can think of it as being a separate gene circuit from the gene circuit that's occurring within the plant.
How do you know which genes to use? So you go to all the corpus of knowledge and read all the research, and some
Yeah,
nerdy guys, they said, like, this specific gene produces, I don't know, many petals. And you're like, oh, I will use it.
yeah, that's exactly right.
Yeah?
That's the fun part. So for the flower engineering project, I don't do really much discovery. I would like to get to that point, but right now it's really more of an art and science communication project. And the big mission of it is to show what we've learned about Arabidopsis and kind of distill that into a really visual plant that people want to learn more about it. So when I usually do public outreach in shows, for example, people have never heard of Arabidopsis, despite the fact that it's the most studied plant in the entire world. There's 50 years of research on it, 50,000 papers that mention this plant, and probably over $1 billion of government funding that have gone into figuring out how it works. And so of those 25,000 genes, we discovered the function of a ton of them. And it's written within the literature. You just have to go find it. So my favorite pastime is just going through the literature and not really fully reading the paper, but just kind of going through the figures, looking at it as like a catalog. And then if I see a phenotype, like the effect of a mutation on the appearance of the flower, if it's quite nice, then I add it to my flower design Illustrator file, and I have a wish list of things that I want to CRISPR together. And yeah, I try and combine together the most aesthetic phenotypes that have been found, plus some other cool things.
Yeah, I can imagine Nick going through his studies and what do we have on the menu? Okay, this looks
Yeah,
nice.
yeah. It's like a wizard's like cookbook for plants. It's amazing. I'm very privileged to be able to have done this project, and it's quite funny. Once I put all of this together and I make this flower, even some of the plant scientists that I talk to, they don't even believe that I've really made this flower because they've only seen it online. The people in the lab, of course, have seen it and they know it's real. But they're like, nah, that can't be really a Arabidopsis, and that's what I find really cool. Because when you're on like the cutting edge of science, it starts to look like magic, because it's something that no one's done before. And yeah, that's a major part of the project is showing what is the power of basic research. Like I didn't discover any of this. I'm just the person putting it together. And it's only possible because we've figured out so much about flower development and all of these other things.
Why this Arabidopsis that many people even have no idea about? Why this specific flower? So you took it because it's very well researched, probably. You have the
Hmm.
most research, so you had a lot of information that you could use for this project, and also because it doesn't look, I mean, anyhow special. So I felt like, what I
Exactly.
can do with that. So yeah, but
Yeah.
why government spend so much money on discovering the traits of like just a normal flower? What's so special about it?
Yeah, right. So you pretty much nailed it. Those are the two reasons that I picked this one. One, that it's kind of an underdog. If you want to make a really extraordinary flower, it's best to start with something that's not so extraordinary. It's like making a Lamborghini out of a Ferrari versus making a Ferrari out of a Ford Model T. Which one seems more impressive? So there's that kind of aspect. And then there's also the aspect that it's the only plant that you can do this on because we know so much about it. There's other model organisms for plant science where we know quite a bit, but not nearly to the extent as Arabidopsis. And there's reasons that it's a model organism. It's the simplicity that makes it not so extraordinary, but it's the simplicity that makes it an extraordinary model to study because it's genetically tractable, transform it, the genome's relatively small, it grows quickly, produces tons of seeds, all of these benefits. And so if I wanted to genetically engineer a new beautiful type of rose, this is going to take on the order of decades, right? Just because it takes so long to grow and transformation's difficult. But as you can see, just within a year and a half, you can really transform a Arabidopsis into something completely different.
And they're all stable. So you have all this amazing peony looking
Yeah.
Arabidopsis all
Yeah.
around.
Yeah, it's not as stable as it may look because the mutants that I work with, they essentially remove the reproductive organs of the flower.
Mm.
Yeah, so it's actually quite cool because then it just produces a bunch of petals, which makes it quite beautiful. And it also is kind of a built-in safety mechanism. They're not producing seeds, so they're not going to escape at all. So it actually makes it quite nice in like an educational setting where maybe I could distribute these to classrooms that can take them on because they're completely sterile. But in terms of breeding, it's quite a big pain because if you can vegetatively propagate the plant, it's okay because then you don't have to go through seed, right? But with Arabidopsis, I do a lot of crosses and transformation, and you have to go through the seed. So I'm propagating them as heterozygotes, where one copy of the gene is correct and one is mutated. And then based on the genetics, 1/4 of the offspring will have both mutated and therefore show the phenotype. But now I'm working up into not just single mutants, but like triple mutants, double mutants, quadruple mutants. And so there then like 1 out of 100 show the phenotype that I want. But I've made some genetic tricks, some genetic circuits that can alleviate this so that when you add a hormone to the plant, it goes from this rose phenotype back to the wild type flower where it can produce seeds again, but
It's
it's a
like a
bit
stem
complicated.
cell. Yeah, like turning a normal cell back into the
Yeah.
stem cell condition. So you can practically use a hormone to erase the changes that you made.
Yeah, I would say rescue the changes that
Uh-huh.
I made. So it's like you break a few genes that cause the plant to lose the reproductive organs, but I have the wild type copies still within the genome, but they're just not active. And with the hormone, then I can activate it. And it doesn't go back to a stem cell niche as I think you might think. The meristem, like the parts of the plant that's producing all the organs, the floral organs, it does have a niche of cells that are kind of in this undetermined state, the stem cell. And that can be rewired once I activate these genes so that it starts to produce the reproductive organs again. So you are kind of like rewiring it and sculpting it in this way. And I find that aspect really cool because if you look at the floral meristem, especially in my mutant background, it looks like the nozzle of like a 3D printer as it's like making these little petal primordia that are then expanding. That's why I see it kind of as a cool art form because it's almost like you're sculpting, but you're sculpting with this kind of self-assembling structure that is the floral meristem.
If you think about it, this is really magic. I
It's not
went,
magic, it's science. Remember.
well, I mean, science indistinguishable from magic, especially the
Right.
science that you do not understand how it functions fully.
Yeah.
Then, of course.
Yeah.
And I loved how you wrote on Twitter that flowers follow rules, so the rules can be rewritten. Can you
Exactly.
talk a
Yeah.
little bit and explain what are the rules that flowers follow?
Right. So when I said rules, I was really referring to this model of flower development that was established mostly in the 90s by some very smart scientists that essentially figured out like the instructions for the flower. It's called this ABC model that describes how different combinations of homeotic genes can direct where and what floral organs are placed as the flower is being developed. So, for example, the Arabidopsis flower, in the outermost whorl, you have leaf-like structures called sepals. Then you have 4 petals. Then you have 6 stamens. These are the pollen-bearing structures. And then you have the pistil, which bears the ovules or seeds. And in each of these whorls, you have different combinations of transcription factors or homeotic genes that are controlling the gene expression of this large, complex mess that I was trying to describe to you. And so these are really upstream of all of those genes. And so when they're turned on, they say, okay, you're going to activate the sepal fate. You're activating petal fate. You're the stamens. And how this patterning, though, is established, why this certain combination of genes is only found in the sepals and petals and stamens and pistils is super interesting because they like antagonize each other. And so, yeah, there's other genes that regulate this. But what's quite cool is these rules, this ABC model, it's very well conserved among all flowering plants. There's expansions to the rules and modifications on the rules, but they all kind of follow this principle of different combinations of homeotic genes. And so my favorite thing to do is walk around the garden where I work and kind of guess like what are the rules that govern that flower and how have they been modified, for example, compared to the ones in Arabidopsis. I hope that made kind of sense.
That's extremely interesting. Tell me what kind of features you can modify in a plant and how far you can go in this process.
Right. That's a very good question. I like to break it down into kind of the sculpting aspect and the painting aspect. If you want to design a flower, you can think about its morphology and you can think about its color and patterning. So I break it into sculpting and painting. In terms of sculpting, you're mostly playing with these homeotic genes, deciding where and what organs are going to be placed along the flower. And then you have a further level of like, what is the shape of those organs and how large are those organs? And for that, you're kind of controlling the rates of cell proliferation and cell growth. So that's kind of the sculpting aspect. And there in Arabidopsis, there's literature for all of those different aspects. Like there's described mutations that rewrite the ABC model. There's mutations that cause more cell proliferation or more cell growth. And so I can hand kind of pick those to sculpt the flower, which I've been doing. And then for the painting aspect, I first like to start with kind of the palette. So if you're going to start painting, you need a palette of colors. Unfortunately, right now I just have pink, which are these beetroot genes that I told you about. They're called Ruby. It's a single transgene. But right now I'm making transgenes that can encode all the different colors. Right now I'm starting with the warmer colors like yellow, orange, pink, peach, and whatnot, as the cooler colors are a bit difficult. But once you have those colors, now it's how do you arrange them along the petal to make a design. So you can imagine stripes or spots or color just around the edge of the petal or the base of the petal, or how do they fade into each other. Yeah, I don't want to go too much on a rant, but those are kind of the aspects of flower design that I'm thinking about on a daily basis.
Stupid question here
No,
on
there's no
this on
stupid
this
questions.
side of a chat. Can you use an animal gene and put it into a plant? As an example,
Absolutely. Yeah.
yeah?
Yeah.
Really? So those are the same kind of genes. So
Yeah,
you can like
there's some genes
cross
that are
them.
very deeply conserved from plants and animals. Actually, I'm working on some of them now. Like some of the immune receptors that are used in plants, NLRs, they're also used in humans like us. So we have homologs. There's a lot of genes like that. But when it comes to color, there are actually some genes that you could clone from yourself and put into the plant and change the color of the plant. For example, we make melanin. There's a gene called tyrosinase that converts tyrosine into melanin. This dark pigmentation kind of goes from light brown to black. And people have used this actually in E. coli to produce really strong black pigments that can be used as dyes. And I believe this can also be placed into plants and be used to encode melanin. So I kind of had a wild idea. I was thinking of putting my tyrosinase into the plant to make it have melanin and to be like a black gothic flower. And then it's like I am a part of my artwork and my gene replicates with my artwork.
This is extremely cool. You should do
Yeah,
that. This is
yeah.
brilliant.
Yeah, have you ever heard of the Rorschach test? Like
Yes,
these
of
ink
course.
blot tests? I thought to use my DEX-inducible system to encode this tyrosinase that can make black pigments to make like Rorschach patterns in flowers and call this like the Rorschach flower set. So put this into like many canvas flowers that are just pure white, and then be able to paint onto them with the hormone to induce these black patterns. And then once I'm gone, like in 500 years, but my plants live on, my little gene is in there and it's like melded with my artwork and my legacy.
Oh my God.
Yeah.
This is mega cool. This is brilliant. Is
I'm
it
glad you had that
your...
response. A lot of people are like, wow, this guy's really crazy.
No, no. I think this is brilliant. Absolutely. It's brilliant both from an art perspective
Mm,
and from scientific perspective. And yes, it
yes.
has this craziness in a good
Yeah, yeah.
sense of a word.
Yeah. Yeah, but I think that's where the art really is. Like if everyone loves your artwork and has the same response to your art, it's probably quite mediocre. Like if everyone just loves it. But the best art divides the audience. That some people love it and some people think it's horrible. So you see this when I posted my post about the flower engineering project. the audience was quite divided in the comments. Some are saying I'm going to start the next zombie apocalypse, or that like I hate nature because I'm trying to design it. And then other people saying this is like the most beautiful art project ever.
Well, but definitely they classifies it as art because art should evoke emotions.
Hmm.
Whatever people think, yeah,
Hmm.
they are not indifferent when they
Hmm.
see that. That's why it's super cool.
And perhaps change people's perspective on how they see life. For me, that's really like where the art lies. So there's two kind of central themes that I want with that. That when people see what I'm doing with the flower engineering, that now when they go through a garden, they look at flowers as inventions from evolution, that they're actually designs, beautiful designs. And then also to evoke this question of like, what is actually beauty? You know, when I wrote this post, actually, most of the negative feedback wasn't about biotech or GMOs or any of that. It was that I called the original flower boring and the new one extraordinary. And people are like, oh, you're not a naturalist. Like the first one was just fine. It was also beautiful. And to be honest, I would agree. I studied that flower for 10 years. I love it. You know, I wouldn't be doing that if I didn't love it. But it gets bullied a lot in plant science because it's simple, you know. And very rarely do you hear the Arabidopsis flower as being called beautiful. So it's like, what distinguishes like a beautiful flower from a non-beautiful flower? Is it completely subjective? You could say yes, but also you see that people agree on what's beautiful and what's not. Otherwise, you wouldn't see the same flowers being sold in the grocery store, the same ornamental flowers having the same economic value. And so I want people to really think deeply about like what is
And I haven't seen Arabidopsis sold in the supermarkets left and
exactly,
right.
exactly. That's what I was thinking. I'm like, okay, if you love Arabidopsis so much, then you should grow it and give it to your grandma. Like, she'll love it.
So we touched a little bit the glow in the dark plants. There is also a company, probably you know, the sensory plants.
Hmm.
Guys, well, ladies and guys who work with smells. Yeah,
Yeah.
so they are engineering smells in plants, which is also very interesting
Hmm.
twist from my point of view. What you can do with a
Yeah,
do you think
totally.
that plants like these ones at some point will become a norm, or it will always be a curiosity, some rarity for nerds or for magazines to publish
Yeah.
some articles or whatever?
Yeah, I don't have an answer to that. I wish I could tell you. We'll have to see how it goes with light bio and sensory plants if there's a big enough market for it. Right now, it seems like the people buying these plants are mostly nerds like me that are super into biotech and they're into the science behind it. Because there's plenty of bioluminescent organisms that you can already grow in a tank. So like do you really need a bioluminescent petunia? You know, for me, the cool aspect is that this was like an invention that this was made. So I think until it's like super bright that it can like light your room and have some serious utility, that it won't take off too much. However, I do see a market for like designer flowers, flowers that have like really unique patterns that wouldn't evolve in nature. So I think what Sensory is doing is mostly like recapitulating volatile pathways to that have evolved in nature to make certain smells that flowers already make. But if you could make a scent that never existed before, you know, or make like a really extraordinary smell that flowers don't already have, there could be a potential market for that. Maybe in like the more luxury markets where you have like a house plant and people love to talk about their house plants, but it has this kind of like story behind it that it's very exotic.
Yeah, maybe you can even order yourself in the future a plant with some specific pattern or
Exactly.
specific
Yeah.
color combination or
Yeah.
whatever. But that's
Yeah.
definitely high end of a market.
Yeah. That's actually where I'm trying to get. So I would love to make like custom jewelry out of the flowers that I'm making now. So they're the perfect size for like a ring and a perfect size for like an earring. People keep saying like, oh, you need to make the flowers bigger. But actually, to me, what's really cool about them is like how small they are, and they're the perfect size for like jewelry. So I'm trying to first like make all the colors, make many different patterns and different shapes, and then if I can find a way of embedding them like in epoxy where they haven't really lost their color or shape, I think there could be a market for this where you get kind of like a one of a kind flower. And this could be maybe more expensive than a diamond because you're like, oh, this is even more rare than the diamond.
I believe that there is a market for that. That
Mm.
sounds very cool, very unique, and very special. It can be a great gift, great present. Absolutely.
But the problem is always getting around GMO regulations, and the flowers that I make have so many edits and so many transgenes and so many patented gene parts that it's kind of like a legal nightmare. So actually, that's why a bit why I'm focusing mostly on art now. So mostly just photographing these flowers and creating artworks out of them. So I'm not super focused on like the scent because you can't capture that through the screen or through a print or whatnot. But I'm in the process of making concept art just to kind of elevate this idea of flower design and hopefully have like a lab slash art studio one day where we sell artwork based around flower design, and the money that we make kind of goes back into making the flowers and into like art and education.
So a closed biological system. You make
Yeah.
plants, then you make art out of plants, then you sell the art and you finance
Yeah.
the plants
Yeah.
again.
Yeah. I mean, hopefully making some technology that we can transfer to more economically valuable flowers that people can get their hands on. So I would love people to like interact with their house plants more. But right now it's just kind of something like, do you like this amount of light? Do you like this amount of water? Oh, it has a new leaf. I would like it to be like, oh, you shine a light on this certain area of the leaf, and now it's changed color. Or I heat shock the flower, and now it produces more stripes or whatnot. So a system where you can really start to sculpt and paint the flower genetically through induction systems.
Is it even possible to make a plant so receptive, not on the stage when it just
Mm,
forms, but
mm.
when it's already an adult and can get a passport?
Get a passport. What do you mean?
I mean, it's adult, it's like it's
Yeah,
already
yeah,
grew, and now you're running around putting a light on it and it changes
yeah, yeah.
colors and everything.
That's a really excellent question. Yeah, because most of the patterning and sculpting happens actually when the plant is small. Once you can already kind of see a leaf, it probably already has all the cells in there. They're just expanding, right? And sometimes they become less transcriptionally active. And so if you have a mature petal, like fully grown petal, and you add this environmental stimulus, sometimes it doesn't respond because it's already mature. So for like the patterning aspect, you have to trigger it when the floral meristem is still developing. However, for some flowers, in my experience, once they're fully formed, they're still transcriptionally active and you can still change their color.
I have an idea to take the RNA, the epigenetics, and to activate some genes that you put in before with a
Mm.
light or temperature or whatever. I don't know if it's even possible.
Like apply the RNA to the plant
Yeah,
or,
just like you put it in, it's not activated, and then at some
oh,
point you
yeah.
affect it in some way.
Hmm.
Yeah, and it turns on, and then it reacts
Yeah.
and changes potentially.
Did you
Who
just have
knows?
this idea
Yeah,
or
I just had this
has it been,
idea.
are you a biologist?
No.
Wow. I mean, it's a great idea. This has already been invented.
Oh, really?
Yeah, there's a thing called a ribose switch where
Oh,
I don't know too much about ribose switches, but it's like a folded RNA that's in the presence of some molecule folds such that it can be translated and then it gets turned on. So there's these kind of switches. And that's nice because it's already transcribed, ready to go. And then you get this response relatively quickly. Because the way I do it, you add something and then it starts the transcription, then the translation. So you only see the effect like 24 hours later. But I could imagine maybe if it's the RNA is already preloaded, ready to go, it could be reduced down to hours.
this is
Great idea.
so.
Yeah.
Thank you. I'm too old to become a biologist, but I love it so much. So
But you
I'm.
seem to know, have like a good basic understanding.
Yeah, because I talk a lot with scientists. So I,
Ah, okay.
especially with biotech scientists, engineers,
Mm.
and so on. So like I collect the information here and there and coming up
Awesome.
with weird ideas.
Hmm.
Tell me, where do you find inspiration for your work?
Everywhere,
Yeah, I
constantly.
see that.
too much. Sometimes people just say like a sentence and then I relate it to my project and then I'm suddenly talking about my flower project
I'm
again.
running away. I need to write it down. Oh my
It's
God,
actually
I
why
have
I
an
was
idea.
late for this podcast because I was having a deep philosophical talk about what is beauty and whatnot. I would say I mostly find it in the garden, walking around, seeing flowers I haven't seen before, taking a close look at them under the microscope, thinking about how could I engineer this. Reading papers, of course, is a big inspiration. Also, just looking at artwork in general. There's a guy, Takashi Murakami, who has made he's a very like modern artist. He's made album covers for like Kanye West and made designs for like Louis Vuitton. But he has this Murakami flower that's very simple. It's just like a smiley face with some petals. But the composition of these flowers that he makes is really thought evoking. And he makes kind of designer flowers where each petal has a different color and whatnot, and evokes some kind of emotion. So stuff like that, looking at artworks. And flowers have been the subject of art throughout all of human history. And so I like to kind of put this project in a historical context, in like where does this fit in terms of the timeline of flower art? Like if you look in the early 1700s, like they're doing a lot of still lifes of flowers, but you just kind of capture them in like one static moment, you know. And so I'm part of my work is revisiting some of those mediums and those styles and trying to implement my like genetically engineered flower in that style, but also take a more modern take where I'm doing this kind of live imaging, this time lapse imaging of the flowers opening and whatnot, to show that these are living organisms. When you see them in a still life, they're just like an object, right? But they're living organisms, and when they're moving over time, we don't really see this because we just see them at static time points. But once you time lapse them, you almost like feel like the emotion of the plant or like how it's sensing the environment. Like for example, when the lights kick on in the morning, you see the flowers like open up very dramatically, and then throughout the day they're doing this circumnutation. And if they're not being fed enough water, they start to wilt and become really sad. Or if there's too much ethylene in there, they just start dropping their petals. And I find it really interesting how like the movement of a plant can evoke like emotion in you. You know, you say like, "Oh, it's sad," and it's like it's not really sad, but it looks like it.
Have you made already a documentary? You need to make a documentary with your time lapses that would
Mm,
tell a story of a plant with all
yeah,
these emotions,
yeah.
hope, grief, love, whatever happens there.
Yeah. There's actually a girl in the UK, Eleonora, who is a ballet dancer, but she's also a plant scientist, and she does ballet like based on plant movements,
Wow.
and like trying to capture like these moments of like the hypocotyl escaping the seed coat, you know, and feeling sunlight for the first time, all of that. But in terms of a documentary, I think it would be super cool to start a documentary now on the flower engineering project because I really see it as like a lifelong project of mine, something that requires like extreme vision and takes a lot of patience and a lot of time. But if we could get that kind of going and document some of the process, I think in like 20 years when I have this whole collection, you could have an amazing documentary where you're like, I had this vision of designs that I wanted to make that seem impossible, and then you bring them to life. I think that would be cool. Yeah.
That would be mega cool. Yeah,
Yeah.
that would be a mega series. I would watch it in 20 years. Please
Yeah.
do that.
Yeah. Maybe I just need to start filming myself and start making it.
I think it will be beautiful. You have a very refined taste in what you are doing, so I'm sure that it would be great.
Thank you.
And asking for a friend, how a person with no experience in plant engineering
Mm.
could start doing something in this field.
Yeah, I get asked this question a lot, and I feel like my answer is always very disappointing. But it's also a real truth that plant engineering requires a lot of expertise, money, equipment, time, and it's not so easy for someone to see what I'm doing, which is built on the shoulders of a lot of other people, as you can imagine. Also, just securing the funds to make this possible, to do it then in your own home. But I do imagine a future where you have like biotech toolkits, hopefully safe biotech toolkits that have safety measures built in. We don't want people playing around with the wrong organisms. That at home you could start to do cloning and plant transformation. I don't think that's too wild of an idea. but you would probably need to make some of these DNA parts already that people can then order and start doing it at home. You probably know this guy, Sebastian Kotsioba. Yeah,
Yes,
yeah.
yeah.
So he's doing it in his house. So it is possible. But this guy's also a genius and also like the most passionate person you've ever seen in your life. So if you're that like dedicated enough, it is possible.
Well, I know guys who create this labs at home for some
Yeah.
like very, very small money. Yeah, like
Hmm.
do-it-yourself labs, very plasmidsaurus
Yeah.
that can help you to sequence whatever you
Yeah.
want to sequence. So who knows?
What I would say to someone that wants to get into plant engineering is first start with tissue culture. Like you can get the laminar flow hood off of like eBay, like a used one from a lab, or you can kind of make your own for varying budget ranges. There's tutorials like that on YouTube. And there you can start to propagate plants in vitro. And this is quite cool. It's like the closest I think you can get to like a lab like experience without investing too much money. And you can start like your own breeding program. I mean, there's 300,000 species of flowers. So you kind of like pick your Pokemon starter, and you're like, I'm gonna tissue culture this. And also when you do tissue culture, you get something called somoclonal variation, where you do kind of induce mutations in the plant just through the in vitro conditions and the hormones you're adding. So you can find phenotypes without doing CRISPR or having mutagenesis.
Well, that sounds like a good place to start.
Yeah.
What are the most exciting advancements in plant engineering that you would anticipate in the
Mm,
next five,
mm.
10?
Yeah. Also a really great question. Well, one of them kind of ties back to the previous question of can we easily do plant transformation without tissue culture and all this specialized equipment? And there's more and more papers coming online of people doing kind of tricks to get the genes into the plant without having to do really complicated procedures. So it's called
Guerrilla engineering.
just the transformation process, which is quite difficult. We often do it in the lab through tissue culture, where it takes a long time and specialized equipment. But you can do it in planta as well, where you, for example, with Arabidopsis, you dip the agrobacterium into the flowers and you can transform it. So that's super simple. People are finding out that you can do this with many different plants. You just have to be a bit creative with it, like where you add the agro, how you add it, if you have it sucked up through the roots, or if you have to decapitate the plant and then inject it in and whatnot. You can even deliver CRISPR now with a virus as well. And so there, you're not even introducing a transgene, you're just introducing the Cas9 that can edit it. So I think this implant transformation of many different plants, like expanding just from the models, that's going to be super exciting, especially compounded with all the genome data that we've accumulated over the last decade. Yeah, we're doing like comparative genomics and then engineering in non-models. That will be exciting. And I also think some of the advancements on Cas9 and the CRISPR system, not just for inducing mutations, but doing like knock-ins, doing prime editing, and even doing whole chromosome rearrangements, stuff like that, where you can actually like introduce whole new mini chromosomes and whatnot, where you're not trying to make new plants by breaking reproductive barriers between species, but rather just like purely engineering the plant by introducing chromosomes, whole chromosomes into those cells. I think that will be quite exciting. Sorry, that
Oh.
answer was all over the place, but you can see I find it cool.
There is a lot of potential here. This is what I see clearly. But what are the next projects of Nick? What are you planning to do next?
Yeah, so I'm very happy to announce that I just got a fellowship, a Swiss Mobility Fellowship, where Switzerland will help me do research in the UK, where I'm focused on plant immune receptors, so defense against pathogens, which is a really critical topic because I wanted to work on something a bit more applied, not just the fun, creative project. And so there I'll be making chimeric immune receptors and kind of engineering the proteins in order to detect different antigens, not just pathogens, but also things within the plant cell, and using those immune receptors as like transcriptional switches so that when they detect an antigen, they trigger a transcriptional response, but also using them for other outlets in synthetic biology. So I was very happy that I got this fellowship. So I'll be doing And I also just got a grant from the O'Shaughnessy Ventures grant program, where they gave me very nicely $10,000 like to pursue my passion on the flower engineering project. And so you'll be seeing very shortly a lot of new flowers coming online with unique patterns, as well as kind of some concept art that I hope to transform into a clothing line based on biodesign.
Wow, that sounds very, very, very impressive. So, how can people stay informed about all these beautiful projects?
You can follow me on Twitter at Nick Desnoyer or on Blue Sky, where I usually post on both. You can also follow me on Instagram as well, but I'd have a very, very small following there.
Perfect. Thank you so much. That
Yeah,
was super cool, very enlightening and passionate. I feel that you are on fire with plant engineering, and this is
thanks.
contagious.
Great. That's the ultimate compliment.
Thank you so much.
Thanks,
Wishing
Maria.
you, yeah, a nice evening.
Mhm.
Yeah. Bye bye.